Hi everyone! We all know that DNA, or deoxyribonucleic acid, is the blueprint for everything that happens inside an organism's cells, and DNA extraction is an essential process in any experiment on genetic engineering and recombinant DNA technology. For this video, we will be extracting the DNA of a banana with the following cheap and easily available materials. Before we begin, we will place our bottle of rubbing alcohol into the refrigerator and let it chill for the duration of this experiment. It is important to note that if the alcohol is too warm, the DNA might denature or break down. Thus, cold alcohol is preferable, and it will help us gather a larger amount of DNA. Additional reminder to wear protective gear like gloves and mask, guys. Also, be sure to handle all materials with care and caution because safety is our priority. For the first step, peel half of a banana, put it in a resealable Ziploc bag, and then close the bag again. On a hard surface like a tabletop, carefully mush the banana in the bag for about a minute until all lumps are gone. We are mushing the bananas because this separates their cells. It makes the cells more exposed to the soap and salt, which makes the extraction of DNA a lot easier. Second step, fill a measuring cup with half a cup of warm water and a teaspoon of salt. Warm water will help dissolve the salt faster. Third step, pour the salt water into the bag and close the bag. Then, gently mix and slosh the salt water and mashed banana together for 30 to 45 seconds. The salt water is crucial because it will help the DNA strands to stick to each other once dish soap helps release the banana's DNA. Fourth step, add half a teaspoon of dish soap into the bag and again, mix around the contents gently. We are doing this because the dish soap molecules will react with the lipid particles in the cell membrane. This will dissolve the cell membrane and allow the DNA to be exposed. Fifth step, place the strainer on top of the narrow glass, carefully pour the contents of the bag into the strainer and let it sit for several minutes until all of the liquid has dripped down into the cup. Remember that we filter the mixture to separate the large particles because we will not be needing them. Sixth step, take the rubbing alcohol from the refrigerator. Then, tilt the glass and slowly pour the alcohol down the side of the cup until there is a layer that is 2.5 to 5 centimeters thick. It is important to know that DNA clumps are soluble in some liquids, but not in alcohol. Thus, DNA does not dissolve in alcohol and alcohol helps in the isolation of DNA and in the formation of DNA clumps. Seventh step, let the two-layered mixture sit for 8 minutes. During this time, the DNA pieces are clumping together. Eighth and last step, stick the wooden stirrer into the cup. Spin it in place so that the cloudy layer spools around it. Then remove the stirrer. The substance that you see on the stirrer is the DNA. DNA looks like a stringy substance. Also, each of the materials played a specific role in its extraction because the soap helped to break down the cell's outer membrane, the salt helped to separate the DNA from other materials in the cell, and the alcohol helped the DNA clump together in a separate layer. Thus, all of the materials are equally important. In conclusion, anyone can extract and isolate DNA with cheap and easily available materials like salt, alcohol, and soap because the guiding principle of DNA extraction remains unchanged. The experiment also resulted in the extraction of DNA from the millions and millions of cells that make up the banana, wherein the DNA looked like a stringy substance. That's all for today. Thank you for watching!